All right, guys, welcome back. All right, so this one is about the muscle. So we are, continue from the cell. We just have a quiz, right? And then we talk about cell membrane, membrane potential, protein synthesis, and the membrane protein. And then we talk about membrane action potential. And then we talk about several excitable cells that generate action potential. And so we talk about the nerves. And with this one, we are going to talk about the other excitable cell, which is muscle cell. There are three types of muscle cells in our human body. We have skeletal muscle, cardiac muscle or cardiomyocyte, and the smooth muscle. So we have three types of muscle. What you need to know there are three, and you need to know what's their features. So the only one that's voluntary control, you can mentally voluntary control, that's skeletal muscle. And also by the appearance, skeletal muscle has this striated structure, striation. And you also see similar striation in cardiac muscle. So these two are striated muscle. You need to know that what muscle are striated, what muscle is not. That's just another quiz question, easy to ask. And so this voluntary control with this muscle, you can move, right? So how does muscle move your body? Because it attached to bone. So you can have skeleton. So you probably can create a skeleton, and you just need to have some string to move it, right? And the same thing in our body, we use muscle. The string is nerve to stimulate the muscle contraction, causing the skeletal movement, the bone movement. So that's why we call it skeletal muscle. So it's skeleton attached muscle, voluntary control. And cardiomyocyte or cardiac muscle, that is muscle of the heart. So that's easy. The only organ that has the cardiac muscle is the heart muscle. So that's skeletal, that's not voluntary. Another not involuntary or not voluntary control is smooth muscle. Where do we find it? We find it in other organs that's not heart, right? Other organ like blood vessels. Vessel can do the constriction, dilation, and so that is smooth muscle do it. Your digestive system, you can move the food, propel it to moving it. That's skeletal muscle, squeeze it. Your stomach can squeeze the food, that's a skeletal muscle. And your large intestines, small intestines can squeeze the food. So that's smooth muscle. let me get a drink. Let me get a water. Yeah, so that's smooth muscle. And so all these are smooth muscle. So digestive tracts, urinary tract, you squeeze your urine, it's not just by gravity, going down, it's squeezing by the smooth muscle. Reproductive system, the sperm movement, the zygote movement, the uterus control, for the labor, baby delivery, uterus control, those are all smooth muscle. Respiration, your trachea, dilation, constriction, those are smooth muscle. So those are smooth muscle. These, what's the feature? It's not striated. It's not voluntary. So that's that. All right, so now you know those three type of muscle. There are multiple quiz questions associated with it. We are going to focus on this skeletal muscle. We will talk about cardiac muscle when we talk about the heart. I think that's the next lecture. We will talk about the smooth muscle when we talk about the vessel. So we have the cardiovascular system in the next lecture. But today we are focusing on the skeletal muscle, and we will learn how muscle contrast in order to get a basic understanding of muscle physiology. All right, so let's move on. So what is skeletal muscle here? Skeletal muscle is the muscle attached to bone. So this is a bone, here is a skeletal muscle. You should have taken the anatomy, you should kind of know our body's different muscle organ, right? And so it has attached to bones. So that's the entire thing is a muscle organ. We have a multiple muscle organ. In this muscle organ, you can see that this is one of these muscle. If you even if in your lunch you eat a chicken or something, you see the fiber, right? Muscle fiber. Those are these muscle fiber. And how do they attach to the bone? They attach to bone by tendon. Tendon is an extension of this muscle membrane. Muscle is organ wrapped by a lot of thin membrane to cover it. That thin membrane will attach to the bone. Yeah, this thin membrane, these tendons are extracellular. So extracellular means like outside of the cell. Extracellular matrix. So those are fibrous protein. So cells secrete those protein and then stay in the extracellular space, and they form the fiber. And so this fiber wrapped around the muscle organ, and these extension attached to the bone. So tendon injury is not uncommon, and it's fixable. You probably know Kobe Bryant. He wants to have the Achilles tendon broken. It's pretty bad. I mean, the entire tendon is truncated. And if you don't know, Google it. You see that video. And so we basically just give it time. We provide the position, give it time. These extracellular protein will bind together, and then we will form the tendon. So that's that. All right. So yeah, and here, so this is muscle. If we look into more detail about this muscle, you can see that. Let's see that. Let me double check if I record it. Yeah, I do. All right, you can see that these muscles are like this. What about this? What are these? What are these? Well, I kind of get it. We kind of get it. This is muscle fiber. All right. So is it warm? No, it's muscle fiber. So this muscle fiber, what is this? So basically, this show you the formation of a muscle fiber. So in the beginning, you have cell, just like every other cells. You have nucleus, you have cell body, and to form a muscle, you need to know muscle fiber is pretty big. That fiber is pretty big. It's visible, and so it's very long. You connect bones, right? So it's very long. It's also very, kind of big compared to other cells. So single cells is not strong enough to form that fiber. Nerve is also very long, but nerve is single cell. It doesn't really sustain that force. So nerve is not combined. But for the muscle, each cell is formed by a merge of multiple muscle cells. So this is the original cell, and then merge together, they become single cell. So you can see that here is a muscle cell, is an elongated, and the cell has nucleus. Nucleus will get pushed into the border of the cell membrane, and then inside of it, you have a lot of protein. You have cytoskeleton. These protein cytoskeleton will form a lot of this string right here, and this string will appear as striated. So, so that is the structure. So what we got here? Okay, skeletal muscle is a muscle connect with two skeleton. With what? With tendon. All right. Should be easy, right? Okay. And then that muscle is a single cell, but that single cell has multiple nuclei. Yeah. So that's that. So let's look at these muscles, see how do they do the contraction? So next slide right here. All right. So this is, we just basically just coming from here to get these cells and present it here. This sorting is a muscle cell. So muscle cell is a compartment, it's an isolated compartment. You have cell membrane, plasma membrane, somebody call it plasma membrane. And in the muscle, we call it, this membrane, we call it sarcomere. It's just a name for muscle cells. We call it sarcomere. Muscle cell, we have exact same organelles or cell parts, we just have slightly different names. All right. So that's a cell membrane, and these are the nuclei, and so they don't take up space in the major portion of the cell. You can see here mitochondria, right? And here is a lot of these strings. These strings are called myofibril. So myofibril is a combination of proteins. This is somebody call it contractile protein. So this protein can do the contraction. These proteins include different filament, two main filament you can see here. So this is the string. It contains the thin filaments and the thick filaments. And these thin filaments and thick filaments are oriented, very organized. So if you like zoom out, you can see that they are striated appearance. So let's see what we got here. So this is myofibril, that's this one here. You have a cell membrane, you have nucleus, you have mitochondria, you have sarcomere, that's cell membrane. So that's that. If you open this region, expand it, you can see again cell membrane. You can see that cell membrane has some holes in it. So there's some tunnel into it. This one is important. We will talk about it later. This tunnel is called T-tubule right here. So T-tubule is a cell membrane and sink to it. So you have a hole. And so everything out here can flow through this tunnel and flow out. So this provides a lot of advantage because now cell is very thick and environment, glucose are outside. So you need to have a good way to distribute those into the cell. This one also provide action potential to travel, travel along the cell membrane. So it can travel along with T-tubule. So when you excite it, it can excite deep down into the center of the cell body. What is this thing? This thing is called sarcoplasmic reticulum. Sarcoplasmic reticulum is reticular, sorry, what's that? What's that? Endoplasmic reticulum. So in the cells, endoplasmic reticulum. So in the cell, we have endoplasmic reticulum, but here we call it sarcoplasmic reticulum. So that's the same thing. What does it do? It has a lot of network in a way, and so there are ribosomes on it, and we have smooth ER, rough ER, right? So that's a sarcoplasmic reticulum. In the muscle, this is important. Not only that it provides the function as an endoplasmic reticulum, it also is a major organelle to store calcium. We will also talk about that. So that is the structure. So this is just sarcoplasmic reticulum. You have the terminal cristina, just a lot of the like network reticular kind of structure. Yeah. So that's that. All right, so let's look at this one because here is how we do the contraction. So we will go into, so we'll see from the cell, look into this structure, and then we look into this structure. So let's look into myofibril. So this is the site of contraction where we do the contraction. So how do we do it? So what do we have here? We have thin filament, we have thick filament, and before so let's just take a look about these two filaments. One is a thin one. So this is a thin one right here. This purple, this pink thing, this is a thin one. This is a thin one. So one thin filament, one thin filament, one thin filament, and between thin filament, you have thick filament. So this is thick filament right here. So thick means filament interlace with thin filaments in a way. So this is thin filaments, is thick filaments. They kind of interlace with each other. So that's that. So if we look at this one here, so this is myofibril, and so it's very long, and it has striated. So what's that striated? Those striated basically are kind of like segments. What do we have those segments? So these segments are basically defined by the Z disc. So this is a Z disc. Z disc is anchor for thin filaments. So here is a thin filament. They anchor onto the Z disc. So Z disc is crossing over the entire muscle. Let me show here. So crossing over, this is a Z disc right here. Z disc is crossing over the entire muscle. So the thin filaments will attach to it right here. Thin Z disc, Z disc here is M9. Z disc, thin filament will attach to it, cross the entire muscle. So that's that. So this is Z disc. Z disc, Z disc, and it has thin filament, all right? These thin filaments interlace with a thick filament in the middle. So that is Z disc, Z disc, Z disc. So we is segmented by this Z disc. All right. So the segment between the neighboring Z disc is called the sarcomere. It's called the sarcomere right here. Yeah. So that sarcomere is the unit of this contraction. So let's look at what is that segment. So in that segment, this is sarcomere. Why sarcomere is just the basic unit? You're gonna repeat this over and over again. This sarcomere is between two Z disc. All right. So in here we have thin filament, thick filament. All right. And then in here we can see that here is a thin filament. Here is a thin filament. Interlace with a thick filament. So here is a thick filament. So you can see that here is a, if you looking furthermore, you can see this is a thick filament. This thick filament, each filament is formed by its multiple proteins. In thick filaments, these proteins are called myosin. So this myosin is a quaternary structure protein. That's why we talked about the protein earlier. Primary structure, secondary structure, tertiary, quaternary. Quaternary structure, you have multiple chains. So combine all together, and all these proteins form these myosin, large protein, these different chains form this myosin protein. This myosin protein is a motor protein. A motor protein has a capability to conduct a mechanical force. So here is a myosin tail, and this is myosin head. So if you look at that here, these are myosin head. Now it's kind of laying down, but it can extend like this one here. When it extends like this one here, they will get in touch with thin filaments. So when the nerve is excited and going to do the contraction, that's why it's called excitable cell. You need to stimulate it. generate action potential, and the muscle get excited. Different cells in the nerve, when it's excited, it transmits the electric signal. For muscle cells, when it's excited, it will cause this myosin head attached to thin filaments. When it attached to it with the ATP as the fuel of the cell, it will cause this myosin head to bend. When it bends, it will move this thin filament. It's very much like rolling a boat, right? So here is a boat, and so you have two sailor in a way to move the pedal, and so it will propel these thick filaments. It will propel this thin filament toward the center. So two thin filaments from the disc, it will, when it's contrast, when it's conduct contraction, muscle will get shorter. The two Z discs will get near to each other. Thin filament will meet each other because of the thick filaments. The myosin head attached to it and pull it. So you have a lot of this myosin head attached to it and pull it, attached to it and pull it. So that's that. So this myosin. So myosin is the driving force of contraction. Myosin is a motor protein. So it will conduct this myosin head banging, and in order to pull the thin filament. Makes sense, right? All right. So and then here is the thin filament. Thin filament all are aligned with this Z disc. This is thick filament. Thick filament is not floating around. They will all anchor on the M line, and also in the terminal of this thick filament, they will also using a coil protein. This is called tendon. We later will talk about that. To here, what's that? Tintin. Let me see here. Tintin. Sorry, tintin. This is a very coiling protein. It will hold on this it. Right. So this is that basic idea. All right. So before we jump in to talk about the structure, let's look at this band. So here is what we see is appearance. We have sarcomere, and from here you can see like light band or I band, and the dark band or A band. So here is the A band. A band is also called dark band, or I band. I band is the light band. So what's the difference? You can see that they are different because in the A band you have a lot of thick filaments. So these thick filaments make it darker in a way, and it causes the light cannot penetrate through very well. So we call it A band. This A refer to anisotropy. Don't need to worry about the detail, but it's called A band. And I is referred to isotropic, so it's very pure. It's only thin filaments, and so that is these two bands. And so that's all you need to know. All right. And then when we contract, when muscle do the contraction, these two Z discs will get close to each other. So the sarcomere will get close to each other. And these light band will also get shortened. So this light band will also get shortened. Everything will kind of squeeze together. The only thing will not get changed is the A band, because A band is fixed by the length of the thick filament. So it's not changed, but it's the other thing will get shrink together. The shortest that it can go is to reach to the A band. Yeah, so that's that. Muscle will get shortened. When you contrast, it gets shortened. All right, so that's that. So these slides summarize all these different segments. Sarcomere, this is a unit of contraction site. A sarcomere is segment of this myofibril between two neighboring Z disc. All right, and then Z disc. What is Z disc? Z disc is a like something that all these thin line anchor onto it. So that make the thin line or align very well. That's why we have this band or striated appearance. So that's the band. So Z disc. And then A band, what's A band? A band is an isosocial base, so it's dark, and is the main components in there is the myosin. And what's I band? I band basically is a region that doesn't have the myosin, and so I band will get shortened, and by doing so, your 2Z disc will also get shortened when it's contracting. So that's contracting, right? So that is that. And the next thing we are going to look is to look at these component. Thick filament is kind of easy. It only has one component, that is myosin. And the myosin is very easy, very straightforward. It has a motor capability, so it will move, and that movement is energized by ATP. So that's why we need to have a mitochondria. We talk about mitochondria to generate ATP. And that mitochondria generate ATP. This process is called oxidative phosphorylation. So that's something we has quiz about, you know? Yeah. And you will see it in exam again. All right, so that's the thick filament. And as for the thin filament, even though it's thin, but it has more components, three major components. You see that it has these bits. These bits are called actin. So it's G referred to globular, is because actin can it's polymer in a way. So it will be connected multiple actin to form skeleton, cytoskeleton, not bone, but cell also has its skeleton. So cell has different structure. So that skeleton is formed by actin filament or a polymer in a way. So it will connect together. So the thin filament, the three components, it has actin, this form the backbone of that cytoskeleton. You also have something, a string wrapped around it. It's very interesting because this string is the reason to block the interaction of myosin head with the thin filament. So this is kind of isolation, this separate it. So generally speaking, your muscle won't just twitch by itself because of this, it block it that way. So this like covering this string thing is called tropomyosin. Where is it? Here, tropomyosin. All right. So that's the second thing. And third thing is these units here. So this unit is called troponin. Troponin has three components in it. We will talk about that. So troponin is the lock. So when the cell is excited, troponin will turn, and to allow everything happens. So basically troponin will turn, and to remove this covering, this tropomyosin away, to expose the thin filament, to expose the actin. Actin is where myosin head will attach to it. So before that lock gets unlocked, this tropomyosin covers it. So this actin cannot see myosin head. But when this troponin binds with calcium, we will talk about that. When troponin binds with calcium, it will turn, and then it will remove this tropomyosin away. So this insulation, this sheet will get moved away, expose the actin, and myosin head has high affinity to actin, so it will bind on it and then do the contraction. So that's that. All right, so here are some more detail. The first thing here is the thick filaments. What's the components of thick filaments? So that could be the quiz question, right? The answer is myosin, just one thing, myosin. So here is the myosin. Myosin has different chain. You have a myosin tail, myosin head. Here is the myosin, all this myosin wrapped around, and this myosin head all exposed and ready to contact with the thin filament, particularly the actin of the thin filament. And so just ready there. If there's no voluntary control, they won't touch it. But if you want to do the muscle contraction, tropomyosin will get moved away and that myosin head attached to it. So here the myosin head has a ATP binding site. So ATP is needed. What energizes the myosin head movement? ATP. And what does myosin head attach to? It attached to actin. So myosin head has this ATP binding site. Myosin head also has actin binding site. So that's the site has a high affinity to actin. So if you don't block it, it's like a magnet attached to it. Yeah. And ATP is always in the muscle. So if you attach it, it will contract. Yeah. So that's myosin head. Myosin. It's all like a hold by tendon to the Z disc. Right here, tendon. All right, so that's thick. Then it's thin filament. Thin filaments have three components. As I showed you earlier, you have actin, you have tropomyosin, you also have troponin. So what do they do? Location and function. Location, actin is the backbone of the thin filament. Tropomyosin, okay, that's actin. What's the function? Its function is form that structure. Tropomyosin is wrapping around the actin. And what's its function? Its function is to block, to separate actin from seeing myosin. So here you can see that this actin has this myosin binding sites. These are the myosin binding sites. So it's covered by tropomyosin. And so during the resting muscle, which are contracting, they are not exposed. But if we are ready to contract it, this troponin has three components: Troponin-I, Troponin-C, Troponin-T. So you have 1, 2, 3, 1, 2, 3, Troponin-I, Troponin-C, Troponin-T. Troponin-I has a high affinity to actin, so it attached to everything, hold on to actin. Troponin-C has high affinity to calcium, so there's no calcium in the muscle yet. Usually calcium level is very low inside of cell. So the ions, calcium is ion. The only ion that has high concentration in the cell, why is it? What's the ions that has high concentration in the cell? Higher than the extracellular space. Why is that ion? It's potassium, okay? That's our earlier lecture when we talk about the cell physiology. So in the cell, calcium level is low, so it's ready to bind calcium, but there's no calcium to bind. And troponin-T is one to hold on to tropomyosin. So these three components, basically just like your finger, right? You have three fingers, and the one attached to actin. The other one ready to receive calcium, and third one is to attach to myosin. So it's right there. And when the calcium binds on troponin-C, it then will cause conformational change, and it will cause this tropomyosin to remove. So that's that. All right. So this basically is like a lock to lock everything on site. It can be unlocked by calcium. All right. So that is it. So here is that. So here is the resting, relaxed. If there's no excitation, if you're not doing anything, want to move the muscle, is resting. The myosin binding site on the actin is all blocked. So myosin head cannot bind on it, blocked by tropomyosin. Which one block the interaction between myosin head and actin is tropomyosin. And when we excite it, go through several processes. You need to know excitation is to generate action potential first. And to generate that action potential, you need to have nerve control. So that's why we talk about the motor neuron before this one. And so motor neuron will innervate to touch on the muscle, release neurotransmitter will excite the muscle cell. When it excites the muscle cell generate action potential. Action potential has depolarization, that's sodium influx, repolarization, that's the potassium efflux. So that action potential then travel, and through that travel, you will have calcium to release into the muscle. When calcium release into the muscle, so it's a sequential. So when you excite it, muscle doesn't contract right away. So we have slow twitching muscle, fast twitching muscle. Some muscle twitch very fast, but some muscle it take a while. So that's why we have slow twitching, fast twitching. It's because that excitation and the contraction has a delay. And so it will take a several steps when calcium influx going in. When calcium coming in, it will bind on troponin-C, right here, troponin complex, troponin-C, and then it will cause a removal of tropomyosin to expose these actin, and then re-expose these myosin binding sites on the actin. So myosin head can bind on it and to cause contraction. So that's that. So that's basically the basic idea about the myofibril and the interaction of thin filaments and the thick filaments and the components of these two filaments. So here just summarize what we just talked about. Resting state, myosin binding sites are blocked by tropomyosin. Acting state, calcium is needed. It goes through several steps. We later will talk about that. Calcium influx binds on troponin-C. Troponin, you have I, C, T, three components. It binds on troponin-C, and then to remove the tropomyosin, to expose the myosin binding sites on the actin. So if actin is now get exposed, myosin head can bind on it. Right. So this one showing you that. Here is troponin. Calcium binds on it, it remove the tropomyosin, expose this actin, and then myosin head with ATP, and when it's used, it will change from ATP to ADP, right? And that breakage will release a lot of chemical energy to energize this myosin head. And so that's how you can bind on it and move it. And then you can have we call this one a sliding mechanism, a slide, just like you're sliding the window, just sliding open. You slide it, and two window will get slided, two close each other, so you have muscle contraction. So that's that. All right. So that is the first portion of the skeletal muscle. Let's take a break, and we will talk about we have not talked about the nerve yet, right? We talked about nerve in another lecture. We talked about muscle here, but the following portion we will combine these two. All right, let's take a break.